So I just want to start by saying that if you guys have no idea what this talk is about, I just spent the last few days pottering around in the field, you know, um running after turtles. And so some parts of this talk might come as a surprise to me as well. Um I uh, cobbled a bunch of slides together, and it's going to be a bit of a ramble. So uh, sit down and enjoy the meander. Uh, I actually want to start by thanking RG. I think he's Raghavendra. I think you're here. Um, a few years ago, he actually started putting together a collection about of how many of us as ecologists and evolutionary biologists had arrived at where we are, and uh, I wrote an abstract for that collection, which I think has been put on ice for now. But and the story of that, the title of that was Finding Philautus. And since then, I've sort of you know thought a little bit about how, and when I was writing that abstract, how I arrived at this particular point, right? And and so. Uh, as you can tell from the title, all kinds of things sort of feature in this meandering path, from grass to frogs and genes and turtles, and I'll try and bring all of that together in uh, some semblance of coherence, but mostly, as I said, of serendipity. Anyway, so when I joined CES as a student in 2002, um, yeah, no, 1992. I There were many options open to me because I joined Professor Sukumar's lab, and he of course worked on elephants, but there was sort of never a primary conversation between the two of us. And we talked about working on various aspects of diversity in the Western Ghats. He just received a MacArthur grant to document diversity, and he thought we could ask some interesting ecological questions about it. Now, um, uh, so in a sense, and you can see the role of serendipity here, my PhD was a blank slate, and which meant essentially that every other PhD student in the department felt that it was their right to come and tell me exactly what I should do with my PhD. So from people in my own lab to people like Prabhakar in Professor Madhav Gadgil's lab, Arun Venkatraman in RG's lab, all of these seniors would sit me down and say, "This is what we think you should do." One of the things they talked to me about really caught my attention, and they said, we've been wanting to work in the Upper Nilgiris for many years, and students have gone out there and tried, and it's just like really remote. There's no field station. Somebody's got to go out there and do some research over there, whatever it is. And as I was interested in herps and frogs and snakes and lizards, I thought this sounded like a great idea. And I went out first to visit in, I think, the winter of '92. My friend, now well-known photographer Sarvana Kumar, was doing a project with Sukumar, mapping the sholas of the region, and he'd walk around and sit on these hilltops with a topo sheet and mark the sholas down. And I went out that winter and I assisted him for a few days. We were staying in this little forest department, little cabin, and we froze our butts off. But as a landscape, it sort of really captured my imagination. And when I went out the first time in winter, it looked like this. When I went out after the rains, it looked like this. I looked like that a few years later. But let's get back to the grasslands. Sukumar was then interested sort of in studying the origin of these grasslands, and there used to be many discussions about this at the time. And what was known was that there were these two theories of how these grasslands had come to be. And in fact, there was a very famous debate in the Indian Forester by two foresters, one British and one Indian, one of whom argued that the grasslands were a climax ecosystem. The other argued that grasslands really had been brought up, had been introduced into the Upper Nilgiris when humans had colonized, and then were maintained through a combination of of fire and frost. Um, and Bore, the British forest, was the British forester in question. Ranganathan was the Indian forester, and for decades, this controversy about this origin of the grasslands had raged, in the entire community of maybe 7 people interested in this, including stalwarts such as Meher Homji and so on, who had written about this extensively. And Sukumar had, was working on, of course, worked on plant dynamics and was very interested in this history, and came upon a way to address this problem with a little bit of serendipity himself. And it's a story he's told many times, when he was working on elephants and was interested in studying how much um, grazing and browsing was constituted their diet. He had actually come upon literature from the agricultural sciences where they started using stable isotopes to study, to tell um, the you could use that to tell the difference between C3 and C4 pathways, and therefore you could sort of determine the amount of browse and grass in the diet of these animals. And in fact, he was one of the first to use that to study elephants. And then it sort of struck them that they could use the same approach to look at the proportion of C3 and C4 plants in various strata. And they did this study of peat bogs. And in 93, published a paper in a very now well-known paper in Nature, which demonstrated that grasslands had occurred in the Upper Nilgiris as far back as 20,000 years, well before the first humans are dated to have arrived there. And further research pushed that date back to 40 to 50,000 years, all of which showed that grasslands were a climax ecosystem in the upper plateaus, in the upper altitudes elevations of the southern Western Ghats. And this was both fascinating from an ecological perspective, but also had management implications, because up to that point, the Forest Department, both from British times and then after independence, treated the grasslands and wastelands. Much of the upper plateau was planted over with tea, and then the Forest Department had planted the rest over with pine and wattle and eucalyptus, large tracts of wattle, which would periodically be they would clear fell and regenerate, and because after all, grasslands needed to be planted over. And so therefore there was quite a valuable piece of work at that juncture. It was also supported by work from palynological studies from Poland, which had been actually done by a researcher in the late 70s or 80s in Pondicherry, but the results hadn't been substantiated. And another of Sukumar's researchers, Indrani, worked on pollen, and all of this really substantiated the relative antiquity of these grasslands. Now, while I was interested in these stories, I had nothing to do with any of it, and I was busy, wandering around looking for herbs. My two closest friends were the horseshoe pit viper Trimerizurus strigatus, the Nilgiri wine snake Ahitula perroteti, featured here, and while I didn't end up studying them, 20 years later, when a student, Ashok Malik, actually did his PhD on these two groups of snakes, it turns out that both perroteti and strigatus are actually are ancestral to all other peninsular Indian pit vipers and wine snakes. So these two guys that I met in the Nilgiris back for the first time in '92 turned out actually to be fairly remarkable members of their respective clades. But it's not something that I knew of then or was thinking of at all. I was very focused on this group of frogs, bush frogs, then known as Philautus. And initially, I was fairly convinced that I was going to do my PhD on the one hand, Philautus on the community of frogs dominated by these bush frogs, and shield-tailed snakes. Now, there were two problems. Problem number one was that shield-tailed snakes are really hard to find, and the only ones that I came across after a year in the field, well, maybe not a year, like my first initial field trip of a few months was like, a handful of roadkills, maybe not even like a large number, maybe 2 or 3. On the other hand, I came across lots and lots and lots of bush frogs. And for the life of me, I couldn't identify them using the taxonomic material that was available at the time. And so I went off to Chennai and met the resident amphibian expert over there, and they had a list of species. But I realized pretty quickly that nobody at the time really had a handle on bush frog taxonomy. That the ones that I was collecting really did not match any of the ones that were in the description. It's a good thing, really, because at the time in '93, I think there were 14 known species of bush frogs in the Western Ghats. I think today there are 60. So through work by various amphibian biologists such as Dr. Biju and of course Vijay from my lab and many others. And so good decision. I decided that I wouldn't spend my PhD working on the taxonomy of this group of frogs, that too from just one single hilltop. And I somehow ended up working on small mammals. I worked on these absolutely adorable rats. It's not a phrase that you might have often heard when referring to wild rats, but Rattus rattus in the wild, very likely to be a different species, currently considered to be Rattus satare, though at the time we believed it was another subspecies, Raorchestes, which is an interesting side tale by itself. But I won't get into that today. These were the dominant species. This was the dominant species in the Shola forests. There was a remarkable pattern of habitat selection or competitive exclusion. Never quite got a handle on which one it might have been. It's the pattern of distribution with the grassland rat, which is known as the mitad, and typically found at low elevation in agricultural fields and in secondary forests. But this creature had somehow found its way to the high altitude grasslands of the Nilgiris. Maybe it dispersed there naturally, maybe it ended up there on some cargo truck, but it had absolutely colonized the grasslands. And fascinating pattern. You never found a forest rat 1 meter out into a grassland. You never found a grassland rat 1 meter into a forest. And it was this sort of remarkable distribution that I studied over the next few years, including the Mus famulus, yes, and even more adorable spiny dormouse, so Platycanthomus lesioris. I promise you I did not have to look those names up. Now, I spent several years studying these, the diversity and population ecology of these rats and mice. I think over a period, my field assistants Maivan and Jalendran and I set out 35,000 trap nights, which is so it's a trap night is one trap set over a single night as a trap night, and so you set 100 traps for 5 days. That's 500 trap nights. We set out. We used to have about 200 traps at a time, and over 2 years we executed 35,000 trap nights, which I think was very proud at the time. I think there was more than all of the other small mammal researchers that had worked in the Western Ghats at the time put together. I'm not sure that my field assistant Maivan thanked me for it every time we had to carry one of those like backpacks full of traps across a hill and set them out in the field. The thing that happened towards the end of my PhD, which I was blissfully unaware of, was that the idea of using genetics for ecological studies was sort of coming into vogue. And the late Professor Lalji Singh who later became the head of CCMB, ran this lab in CCMB in Hyderabad, looking at the genetics of wildlife, and they'd studied. At the time, they'd already studied crocodiles, and they were beginning to study other species. And he conducted a workshop in 1997, or maybe it was 98, on wildlife genetics. And my good friend Rana, who looked like that, uh, had attended this workshop, and then shortly after visited me in the field. And of course, you're in the field, you're having endless conversations over these glasses of warm tea in the evenings. And in the course of one of these conversations, I was talking about how my populations of forest, you know, rattles were uh, the demographically much more discrete in the in landscapes where there were many small patches, and they all followed their own sort of demographic cycles, whereas the that I was studying in a larger forest patch, which was where I had multiple plots, but which were inside the same larger forest patch. They all seemed to be much more synchronized, and in fact, that was one of my. In fact, that was the first paper that I wrote. And Rana said, I just went for this genetics workshop. You know what? If they're demographically not connected, perhaps we can find genetic patterns to substantiate this. And I was like, yeah, genetics, schmenetics. I mean, I'm not interested in that. But for those of you who know Rana, you know that he can be persuasive and that he won't stop talking till you agree with him. And Rana persuaded me to collect blood samples from these rodents, and which on my very last field trip I did. I was anesthetizing them with the wrong anesthetic, and ended up killing the first few. But we became absolute masters of knocking a rat out in 10 seconds, taking a blood draw from its heart, and all sort of learned, do-it-yourself style, and collected a whole bunch of rat blood samples. And during the latter half of my thesis writing, I head off to Hyderabad, and Rana, of course, came with me. We'd set this up with Lalji Singh, and one of his research assistants, Shankar Narayanan, who's uh, faculty there now, helped us out, and the three of us would, work through my entire set of rodent samples. I think, in fact, one of Sukuma's elephant researchers, who's working on one of the first to work on elephant genetics, Trupti, was there as well, and we were a little cohort there that was working on rodent genetics together. It was a battle of fun. I learned to do these very high-tech things like use a pipette and so on. Nevertheless, super exciting. 1998, I think it was. We extracted DNA from these rats, and we were doing DNA fingerprinting, and of course the resolution of those markers, we didn't realize it at the time, was too small to actually pick up significant differences. And that project, kind of like, it was this little side thing that I did. It was never intended to be part of my thesis, and I set it aside and forgot about it. But in the course of that work, I'd of course gotten some experience of genetics, and made friends with Lalji Singh and so on. I finished my PhD a year or so later, and I decided that having worked on the hills and on small mammals for a few years, I wanted to go back from the mountains to the beaches. Uh, this has a story of itself. Now, that story starts actually probably around the time I was born. Well, that has to be true in a manner of speaking, but independent of my own childhood. Um, there was this gentleman who did his engineering at the Indian Institute of Technology in Madras, Satish Bhaskar, seen here posing with a garland of sea crates. It's one of those images with which you put the disclaimer, kids, don't try this at home. And this guy spent his day pretty much running to the beach and swimming and running back. And really was quite uninterested in engineering. Luckily for him, Ram Whitaker had started the Snake Park and Turtle Walks along the Madras coast. And to many a college bunking um, youngster, the Snake Park was a refuge where they'd go and hang out and hide away and watch these snakes and turtles and so on. Uh, Shekhar Dattafri, the filmmaker, Harry Andrews, the longtime curator of the Madras Crocodile Bank, um, Preston Ahima is one of the founders of WWF, many others. And as all of them, say, while they were hanging out, this tall, lanky white guy would walk up to them and say, "Hey, man, you want to clean out a snake pit?" And sort of that was the beginning of the end. Something like that happened to Satish. And Satish spent the next 20 years of his life, um, you know, visiting and walking every turtle nesting beach in India, from the furthest reaches of the Nicobar to the Lakshadweep, Gujarat and Orissa, and also to places like Papua and Indonesia. Satish was an inspiration and a mentor to many of us. We were college students at the Madras Christian College and we knew this history of how Rom and others had started these turtle walks and set up their hatchery in a friend's house. And different organizations had run this from the 70s to the 80s. When I was in my third year of my zoology degree at MCC, the forest department, for reasons best known to them, decided to shut their hatcheries down. One wildlife warden is believed to have said the Bay of Bengal has reached its carrying capacity for turtles. We have received so many hatchlings, we cannot release anymore. So there was a vacuum, and something will fill a vacuum, even if it's a bunch of young idiots. And somehow we got it into our heads that we would start a students group and set up our own hatchery. One of my friends borrowed money from his family, and we went ahead and established this hatchery in 1989. The long story cut to the end, the SSDCN, which we thought would survive for a year, 2 years, 3 years, has actually been active on that, on the Chennai coast for 33 years now. So that I think is pretty cool. We looked like that. And like I said, we really were a bunch of young idiots. And the woman in the middle, the girl in the middle, bailed us out by doing signature campaigns at these rich kids schools and getting these 11-year-olds to raise enough money to pay everyone back that we had borrowed money from for this absolutely high-tech palatial hatchery that we built. So we were all, from those college days, I personally and many of my friends, we were by sea turtles of all kinds. We're dying to go and see our first leatherback in the Andamans, green turtles, hawksbills. I mean, it was just like this. The stuff was like it was the stuff of legends for us, and of course, we were also interested in it from a conservation perspective because we knew the turtles got caught in fishing nets and so on. But the other thing that we were really dying to do is go and see a turtle, a Ridley arribada, or a mass nesting event, which we knew occurred in Orissa. So from the 80s onwards, the dream was see a leatherback in the Andaman, see an arribada in Orissa, and so on. And in '97, when I was probably starting to write my thesis, there was this meeting that was organized in Orissa by the Marine Turtle Specialist Group of the IUCN. And a researcher who had worked on olive ridleys in Costa Rica decided to convince the leadership at MTSG that this workshop, the South Asia workshop, or the South and Southeast Indian Ocean South Asia workshop, should be held in Bhubaneswar. Like any good researcher, she had an ulterior motive, and she'd been tracking these ridleys in Costa Rica. She was the first person to put satellite transmitters on male and female olive ridleys, and she wanted to track ridleys in Orissa, and she thought if she organized a meeting here and met government officials, she'd eventually get a permit to do so. She was wrong, of course, but my good luck, they decided to organize it here. This is a story of coincidences. The researcher Pam Plotkin worked in Dave Owens' lab at Texas A&M University. The year that I got into CES, I had applied to Texas A&M, and my application had been transferred to Dave's lab, and Dave had written to me saying, I have funding and I'd love to take you on as a student, but I can only do it next year. And so I had the choice of like going and joining Dave's lab a year from them and joining CES, and uh, split second decision of a sort. I decided to stay. But as fate would have it, uh, I was connected back to Pam Plotkin and Dave again many years later. And I wasn't actually invited for this workshop because I was some guy who was working on small mammals in some institute in India, and nobody really knew who I was. But I heard about it, and I knew that a whole bunch of really well-known, sea turtle biologists and conservationists, names that we'd sort of like read in these papers and absolutely idolized. These guys were coming out to this meeting in Bhubaneswar, so I'm not quite sure what I told Sukumar, but I snuck off, caught a train and, went out to Bhubaneswar, stayed in the house of Devash Pandav, who was making quite a name for himself at the time as a sea turtle biologist. He was doing his PhD on olive ridleys in Orissa. Um, and we went out to Gahir Mata to, um, look for the arribada as part of the workshop. Didn't see a damn thing. But by the time I came back, the talks that I'd heard at that workshop had really inspired me to consider working on turtles again. The range of different sorts of biology that was being done, its implications for conservation, all of it was really attractive, and I'd kind of started. I'd started moving in the direction already of when I finish my PhD, I'm gonna explore this world of sea turtles, which I hadn't really had an opportunity to do earlier. Now, one of the things at the time that in the 80s when we ran the SSTC, and we'd go out and give talks in schools and talk about, this is a sea turtle's life and these are the threats, blah, blah, blah. And it had been hypothesized from the, in the scientific literature, I thought from the 70s and 80s, that sea turtles come back to the same beach where they were born, when they become adults. And this was based on a couple of on the fact that you often found adult turtles coming back to the same beach. Now that history goes back much further. The first known record that I found of tagging is of a British officer in Sri Lanka who put metal rings on a hawksbill turtle in 1794, ostensibly to test that theory of turtles coming back to the same coves, as he writes. In the mid 1800s, there are naturalists who write about the phenomenon of turtles coming back, of turtles undertaking very large oceanic migrations and coming back to the beaches to nest over and over again. Now, by the 70s, folks kind of knew anecdotally that this happens, and some early tagging programs had shown that the adults return to the same beaches. But how? And so there were two hypotheses. One was social facilitation, that is, turtles at breeding grounds followed other adults. And the other hypothesis was that they came back to the beaches where they were born, and there was even a theory about how, due to continental drift, beaches that were closer together may have been pulled apart, and that's how these long distance migrations had come to be, and so on. The problem was that, as you can see here, the hatchling is yay big, like the size of my palm or less, depending on whether it's a Ridley or a leatherback hatchling. But they grow from that size over a period of 10 to 20 years, from literally a few grams to 50 to 60 kgs in the case of a Ridley, 500 to 600 kilograms in the case of a leatherback. From a few inches to a few feet, or 6 to 7 feet, as in the case of the leatherback. Fine. Problem number one: you got to put something on it that will stay across that size, amount of growth. Problem number two: a turtle biologist had written a very crude, done a very crude demographic analysis in 1985 and 87, and written two papers. Proposing on the basis of his calculations that only 1 in 1,000 hatchlings survives to adulthood. One of the remarkable things is that we still say that these days, 1 in 1,000 survives to adulthood. That there hasn't been actually a whole lot of research since Nat Fraser's work. Nat sadly passed away a few weeks ago, and left us this legacy of this 1 in 1,000 hatchlings. No one knows if it's really true, but uh, we believe that it's not far off by in terms of orders of magnitude. Now, the number of tags you have to put on a hatchling, the kind of tag that it stays to adulthood, and the kind of PhD student who studies this, who waits 20 years for the turtles to come back in order to prove that these, hatchlings come back to the beaches where you're born, really seemed like a bridge too far in the late 80s, right? So we would tell the story to the students, and we'd be like, "This is how it is, and this is why we don't know whether they really come back or not, and one of these days we'll find out." Right? And we all, I mean, I think like everybody else, I assumed it would be some kind of fancy tag that would give us the answer to this question. But no. In 98, when I was beginning to conceptualize what my postdoctoral proposal would be about, I started reading the papers, and I was really annoyed when I discovered that uh, turtle biologists had started using genetic tools to answer this question. And essentially, what they were doing was when with populations that had uh, uh, you know there were large known feeding grounds and multiple nesting grounds nearby. They were assessing the genetic stocks using mitochondrial DNA haplotyping at the time of the feeding grounds and of the nesting grounds. Uh, this image actually doesn't show that particular study, but a classic study in the Atlantic studied green turtles that fed off the coast of Brazil, nested in Tortuguero in Costa Rica to the north, and an Ascension Island, which is one single island in the middle of the Atlantic. And you got a whole bunch of different haplotypes in Brazil, but the ones in Ascension Island and in Tortuguero were unique. So basically, if you got A, B, C, D at the foraging ground, you always found A and B at Ascension, and always found C and D at Tortuguero. Suggesting very, very strongly that uh, unless there was natal homing, unless these hatchlings that emerged from these nesting beaches came out and fed in common areas, unless they were going back to their natal beaches, that you would not see these genetic patterns. And so they were using um, genetics to study population structure, evaluate natal homing, and so on. And again, as luck would have it, um, uh, Brian Bowen, who's sort of one of the pioneers of sea turtle phylogeography and has now spent the last 20 years working on sharks and other marine fauna. Brian was one of John Avies' student. John Avies, of course, sort of being the phylogeography. Uh, I met him at a conference. He was very encouraging, and one of the areas that he had failed to sample in his global phylogeography studies was Orissa. And so Brian was very happy. He said, "Oh, that's great. If you want to do a postdoc, I can't actually find you any money, but I can give you all the moral support that you need." But that was great. Uh, I contacted BC Chowdhury at the Wildlife Institute, and we started our work on the genetics of ridley populations in India, and subsequently um, other species as well. And I think all of this was amongst when we collaborated with Lalji Singh's lab. I was already familiar with the lab, so it was like, "Oh, great! I know how to do DNA analysis. I can work a pipette without breaking things." And so I went back to CCMB with a project from WRI. Over the next few years, we studied ridley genetics. We found that the population of the east coast of India was ancestral to the populations of ridleys across the world, and came up with theories about how that might have happened, that that climate change might have influenced it 4 or 5 million years ago. And interestingly, that the Atlantic and Pacific may actually have been recolonized multiple times from the Indian Ocean, sort of an evolutionary sources and sinks scenario. That sort of led to my research on various aspects of sea turtle biology over the next 10, 15 years in Orissa, mostly at the southern nesting site of Rushikulya, where we've been documenting arribadas since 2007, and are beginning to show that they're stable or increasing, which is all nice and fun by itself. Eventually got around to tracking some Ridleys there and showed that they kind of like pretty much wander all over the Bay of Bengal uh, as we expected. Um, a side story, we were also as part of the genetics project. I was also went out to the Andamans for the first time. Uh, we had a monitoring camp at Galathea on the east coast of Great Nicobar Island, and an island that was unfortunate, a beach that was unfortunately washed away by the tsunami in 2004. Unfortunately, the researcher that we had stationed there at the time died. But in a remarkable story, our field assistant survived a 20-meter tidal wave, was washed out to sea, spent 10 days at sea without food or water, managed to make it back to shore and survived. Um, you can read about it in a story that's been transcribed in current conservation. Uh, that's me back then. Moving on. We also tracked a few years later leatherbacks from Little Andaman, where my researcher Adit has been maintaining the camp for the last 10 years. And as you can see here, some of them go east all the way to Western Australia, as people thought. Everybody said our turtles come from Australia, but then turtles have made it their life's mission to prove turtle biologists wrong at every turn. So the first 8 turtles went out, the first 7 turtles went out east, and we were convinced there's a pattern. As you can see, those tracks all move roughly in the same direction. 2 transmitters bombed. I mean, they just like um, uh, uh, didn't transmit a single signal. The battery failed instantly. So we wrote to the manufacturers and said, we're these poor Indians, can you please send us replacements? They're really lovely guys. And they said, not only will we send you replacements, we'll send you a new upgraded model. And so we got these 2 extra transmitters that we didn't think we'd have. And as I said, turtles are out to prove us wrong. Those 2 guys went west. One went to Mozambique and the other went to Madagascar. Now, while all of that was going on, somewhere in that time frame, this is not entirely chronological, I had come back to CES, and while I was, I left CES primarily as a sort of ecologist and someone interested in community ecology. Through this work on the on the evolutionary history of sea turtles and sea turtle phylogeography, I started using uh, genetic tools quite a bit. And in fact, there was another collaboration with CCMB and others that we were looking at, frogs that were sort of somewhat involved in we were also working on um, the purple frog Nasicobatrachus at the time that SD Biju published a paper on it. I was already getting a little bit interested in the evolutionary biogeography of frogs. And like my character Tavalai in Philautus Frog, my students and I were out on this big adventure, which culminated in several PhDs, of course. Varun's on Nyctobatrachus, Ashok's on various species of snakes and so on. But one of my first students, Vijay, focused on bush frogs, and for me, in a way, it was like coming a full circle because this was an organism that I really wanted to study as a student. Uh, Long story short, over there, Vijay discovered many new species. You know, Published a very nice paper on the drivers of diversification in these bush frogs, from Pleistocene glaciations to the role of barriers such as the Palgard Gap, as well as environment, and how all of these lead to speciation in different ways. Um, Now, one of the interesting things that, well, and um can't give a talk about bush frogs without showing a pictures of few. And at the end of it, there was this really remarkable radiation of bush frogs in the Ghats. Now, the interesting thing about this, and um, some of this, of course, is in the paper that he wrote, and other parts of it are not. But one of the most fascinating patterns was the difference in the the broad scale morphological, the adaptation of these frogs to the environments they were in. Now, the northern clade had these open canopy frogs, um, had a much higher proportion of them, and the southern clade had a much higher proportion of closed canopy frogs, which sort of reflected the habitats in which they had diversified over the last 15 to 20 million years. In fact, the northern clade does not even have, sort of middle and uh, upper story canopy species. Now, the northern Western Ghats, which has a much higher proportion of these frogs were also morphologically were quite distinct in the sense that they had adaptations that were uh, uh, um, suited to these open canopy habitats, primarily grassland habitats. And even behaviorally, you can see these frogs, walking on these, in these grasslands among the grasses. And this is a divergence that took place, over 15 million years ago. And there is dispute about, well, not a dispute, discussion about when the first sort of grassland adapted frogs might have evolved. But it's certainly on the scale of, millions of years, perhaps even more than 10 million years ago. And that then brought me back to that story about the origin of these grasslands. And we, now discussed it, and we said, oh, wait a minute. We can show using some sorts of evidence that grasslands go back, 50 or 100,000 years. But if some of these species evolved 10 million years plus, then there must have been grasslands here then. Were they the same grasslands? We have no idea. But that also coincides with the sort of the global expansion of grasslands as a biome, which is dated at about 15 million years. And so the presence of these frogs in these higher elevations indicates that these grasslands might actually have evolved, that the grasslands and the frogs might have, evolved, appeared and evolved there at the same time, which to me was a fascinating return back to that story that I wasn't actually directly involved with in any way, but had always sort of like fascinated me, and sort of remained at the back of my mind. But the grassland story apart, I think I'm more or less done. The grassland story apart, as the rest is history. I mean, I'm not going to sort of dwell on the research that we've done in the lab. Genetics became a core part of what we do in terms of our work on evolutionary biogeography, and so with all of the students that I mentioned and various other researchers that have passed through, we worked on I think probably like 8 different genera of frogs, uh, 8 different families of frogs, multiple families of lizards, several genera of snakes. And it's really been, apart, I think um, I'm not a taxonomist. My colleagues, I didn't want to mention in the passing. I did that the work that the antiquity of the grasslands has also been supported by other work that people like um, Ishan Agarwal, who's Praveen's student, have subsequently done um, on geckos in open habitats at lower elevations and so on. But many of us who've run labs that have elements of phylogenetics and biogeography in it have been dragged into systematics and taxonomy, kicking and screaming perhaps. But it's one of the things that many of our students and postdocs in Praveen's and my lab end up doing. The underlying stories of phylogeny, which you heard, the history of the fauna of India, the history of the earth, the kind of questions that Praveen talked about, diversification of the Western Ghats. These are the stories that really are the questions that really fascinate us. But along the way, the side story of these really amazing discoveries have also enriched our lives. And some of you may of course may be well familiar with some of the work that's come out, but I'll Prohetula, which is a distinct vine snake, which is actually ancestral to all other vine snakes. Uh, you know, um, in the world and that are distributed across South and Southeast Asia. Um, Astrobatrachus, which is a species currently belonging to a subfamily of Nyctibatrachidae, to which Nyctibatrachus already belongs, um, is another the genus that we found recently. But the lovely thing about all of these is they raise more questions than answers. Why does Astrobatrachus have a single species where a very, very close relative, Nyctibatrachus, has like, 40 to 50 or more? So through all of this, I think all of these threads that came together from, grass and genes um, and put the right interpretation on both grass and genes, if you will, and frogs and turtles. I think have come together in really uh, valuable and interesting ways that I would never ever have imagined starting out. Uh, so with that, I'm gonna end the talk, end my rambling, uh, and say thank you, and allow serendipity to enrich your science. I'd be happy to take any questions.
Thank you, Kartik. I think we already have one hand raised. Yes, Praveen, please go ahead.
Yeah, Kartik, nice talk. Yeah, thanks. Hey, it's the pro Hitler story. I wouldn't really call it ancestral to all the vine snakes. I would just call it sister, because it is just sister, right? And it might have retained some ancestral characters, but I think calling it ancestral.
I'm not taking any questions on evolutionary history today. Only my history. So I won't comment on that, but we can pick up that conversation later.
Yeah, sure. Anyway, yeah, your history was really interesting. Yeah, it's all this amazing stories, and it sort of overlaps a bit your story and mine, because I was also in Lalji Singh's lab.
Absolutely,
I was just thinking about that, and then while you were in WII also, that I was there for a bit, and it's just interesting how things pan out. Anyway, thanks for the talk.
Yeah, thanks, Praveen. In fact, let me say that if I'd had more time to put together this talk, I would have put pictures of Praveen visiting Rishi Valley as a 20-year-old, and as a young birder, and that's the first time we met. He was, I think, interning in RG's lab at the time, and then 6 years later, we were in CCMB at the same time. Overlapped in WI when you came for a conference, and we started talking about the frog genetics. And and then we ended up in CES together. So we've gone bump, and then, and then yeah.
Anyway, thanks. Wonderful talk.
Yes, Raghavendra.
Hi, Kartik. Thank you. I enjoyed it. My question is, can you hear me?
Yes, absolutely, yes.
Serendipity notwithstanding, can you make any predictions of the next stop in your journey?
Oh, absolutely. When I finished my directorship and came back to IISc, one of the things I was saying is that I feel like the 3 years allowed me to wipe the slate, not wipe it clean, but relook at what I might do. And in fact, I was telling my students, everyone who listen to me, I'd say I feel like a PhD student. I can come up with a proposal and do anything that I'm trained. I mean, perhaps not theoretical physics, but anything that's within my capability. And then of course I paused, and at one point I told someone, except I can't lose my job. At which point somebody christened me as annoyingly happy. So but that story apart, I actually again through a little bit of serendipity. Siddharth Sharma crossed me one day on the road in IISc and said in 2010, and said, I'm taking my LTC and going to Lakshadweep to learn how to dive. And I said, you can't learn to dive before me, because I work on turtles and I've never seen a turtle underwater. So last minute, I fixed up a trip and went to dive just so that I could learn diving with Siddharth Sharma. But it just became, that then became a thing for me. And over the last 10 years, I mean, I'm sure all of you know that I've been spending more time in the islands and spending more time on in water research. And that is one of the things that I very much plan to do. My interests in community ecology and biogeography remain, but I plan to focus on reef systems and probably reef fish. That's kind of the current thinking.
Thank you.
I'm sort of like I'm semi-retiring from turtles. I've been making that announcement because I don't think I can actually take on another thing without clearing some things out. So I'm passing the long-term monitoring on to the to Muralidharan and Orissa and Aditya in Andaman. They've been managing it for over 10 years, and so by the end of this year, I'm hoping to hand over to them and yeah, move on to fish.
Great. Good luck.
Thank you, Arjun. So I'm happy to take any questions, guys. I was just like this. That was just for Praveen. I mean, so from the rest of you, I'm willing to take.
Hey, that's not
the topic.
Hey Kartik, I had a question. So these frogs that you see in the grasslands, are they also found in other grasslands of India? I'm not like, hi. I mean, or are they very specific to the sholas only?
Yeah, actually, so most of Western Ghats bush frogs are fairly narrow range. Some are more widely distributed. Off the top of my head, I don't think any of the grasslands are the mountain ones. So the more widely distributed bush frogs are actually the middle elevation, low elevation species. So I don't think there are any widely distributed grassland adapted, open canopy adapted bush frogs. You get other open habitat adapted frogs from other families and genera and at lower elevations and even across peninsular India. Other genera that are primarily dry habitat specialists. But the ones that the group that I was talking about are pretty mountain specific.
Okay. Cool. Thank you.
Hey, Kartik, I had another question. I'm not able to raise my hand, but can I just go ahead and ask you the question?
Yeah. Sure. Go ahead.
Anyway, hey, so I was always, see, bush frogs, earlier we used to call them Philautus, right? The whole group from Sri Lanka all the way to Southeast Asia.
Yeah.
And it is, as far as I know, it's a monophyletic group, as it was earlier described. Now, what is your take on it? Why did they split it into so many genera? And do you think it's good taxonomy to do that? Because here Philautus was always monophyletic, as far as I know, and yet they broke it up into so many genera.
So I think one of the principles for that amongst with taxonomists, of course, is finding characters by which you can actually distinguish one genus from another. So that's a sort of at a practical taxonomy level, but also I think that the principle of comparison, like we do for species, right? You use a particular yardstick of we use 3 to 5% divergence on a particular gene, within a particular clade, because that corresponds to well-known, morphologically well-distinct species within that group. So I think using those distances as a measure, those are the criteria that folks may have used for distinguishing them into sort of separating them into different genera. I'm not very I don't know the details of the argument over there, but also sometimes I think it is helpful in the sense when you have these geographically really widespread groups, and within a group that's spread across all of Asia, if you have biogeographically distinct sort of sets which are genetically separated into monophyletic clades, I think that there's a even in terms of practical usage, I think it's kind of fairly, as long as it satisfies the other criteria, it's fairly useful. So I know that this Raorchestes is largely an in situ diversification of the Western Ghats. And there've been out migration. There are species that have migrated out and have dispersed in as well, right?
And Philautus is in Sri Lanka.
And so you get Philautus here, and so you've had, and so the two Raorchestes clades, for example, I mean, I wouldn't advocate it necessarily, but a future taxonomist might actually want to say, like, these are two very distinct evolutionary clades. Sure, there are some representatives of the South and the North and the North and the South, but they're so distinct that I wouldn't be horrified if a taxonomist in future said that the Northern and the Southern clades should be different genera. I'm not suggesting or supporting it, but that's the sort of like, because there's a geographical evolutionary basis for that divergence. There's morphological criteria, and it also then helps with sort of like identifying that name with a particular group and region. Because I could say Philautus and nobody would know what you're talking about, right? Then you have to qualify it so much more.
From what I've heard, for example, Pseudophilautus and Raorchestes, it's very difficult to distinguish the two.
Absolutely.
But I see your geography argument. I think it makes sense. If you say Pseudophilautus, then okay, it is mostly endemic Sri Lankan radiation with one back dispersal into India or whatever, right? So, but it sort of from geography point of view, it does make sense.
A few dispersals. So the other thing that comes into play over here, and I'm not sure if it happened in the Philautus case, is that like with the Psamophilus calotus clade, when we discovered that Psamophilus is nested within calotus, we had two choices. We could have also just renamed Psamophilus as calotus and sort of like had the entire monophyletic group called calotus, right? Now I can tell you that every time I passed some PhD students in the corridor, they would be like, please don't rename our study species, I was surrounded by labs that work on Psamophilus dorsalis. Imagine, like, sitting in that lab and being the guys who changed the name of the model animal. Okay, so but jokes apart, that was one option. The other option was to recognize the Western Ghats calotus as a monophyletic group, Monailosaurus, which of course we did, with your postdoc Deepak. And taxonomists want to rename everything, right? But then from a point of view of consistency, in my discussions with them, I was like, sure, we can either name everything calotus, or logically, we can't leave Psamophilus as Psamophilus by itself in the middle of that group, because we don't have a monophyletic genus, right? And so then the option was to then make calotus monophyletic by treating just the South and Southeast Asian ones as calotus, which may in future get subdivided. Then Monailosaurus is the Western Ghats radiation of it, and Psamophilus is the one that's sitting, as a monophyletic group, related to Monailosaurus. Now, why would we do that? Not just because of these PhD students, but it's so remarkably morphologically different that it seemed, even from like a intuitive naturalist's perspective, why would you take something that is Psamophilus, whether it's dorsalis or something else, so obviously blindingly obvious, and rename it as calotus? Just did not seem right. And so preserving the name of Psamophilus was what, then we said, okay, let's take these Western Ghats guys and rename them as Monailosaurus, and then everybody's fine, right? So sometimes I don't know if these are exact sort of, I don't know if it's an exact science.
So there's also Brachiosaura that is nested within calotus, and it's a ground dwelling gamete, and we sunk it into calotus. The other thing that people do is call it a subgenus, but I mean, this is like a big question. Do you completely ignore the morphological change that has occurred along that lineage? Anyway, I think we should definitely have another discussion on this. this particular topic about
Happy to.
molecular data and whether it is helping taxonomy or causing more confusion. But anyway, I'll shut up. Hope, I think others might have questions.
There's a message from Maria in the chat box saying, please don't rename Philautus. So, geopolitical considerations. Maria, do you have a question?
No, I have a comment. Don't you guys have, when you build your phylogenies and decide what to call things, you have multiple levels, right? You've got geography, you've got genetics, you've got morphology. Then maybe you now need a new column. That's what do your friends want you to do. So the politics would be nice.
Yeah. So in fact, the Brian Bowen, who I mentioned in the earlier part of the story, he was so frustrated with people trying to rename the East Pacific green turtle as as a new species. He and his friend wrote a paper in Conservation Biology in 2001 where they, I think, tongue-in-cheek proposed the geopolitical species concept. I don't think anyone.
Absolutely.
Yeah. And Raghavendra, you'll find this interesting. That the subspecies is called Chelonia midas agassizii, and given the recent sort of furor over Agassiz's views on racism, there's been a mad sort of debate in the community about whether we should drop that name from for that subspecies. It would have been a lot worse if those same people had succeeded 20 years ago and got it named as a species of itself. So
I don't mind using political considerations for new names, but if you bring that in for changing old names, you are in real trouble.
Exactly. What all will you change?
Yes.
So somebody looked it up and found that there are glaciers, mountains, buildings named after Agassiz. So they were like, okay, what and all are you going to change now if you change?
Yes, but consider many more names.
Many more names, exactly. Are there any other questions from any younger people?
There's one.
Sean.
Sean.
Yeah, hi Kartik. Thanks for your talk. So I had a question. So a lot of your interests have been shaped by natural history, observing natural history. So from your history that you presented. So I was wondering, what are your thoughts about the role of natural history in ecology today and shaping the science?
Yeah, I think. Thanks, Sean. I mean, I think most of you know that Sean's my student, and I'm hoping that's not a trick question. It's like, Kartik, you publicly said I have to do more natural history, so I'm not coming back from the field or something like that. But very seriously, I think I'm going to sort of invoke Raghavendra. I'm going to invoke you again. I think in the early 90s, many of you, you trained us to be sensitive to natural history, but to go beyond natural history, because what had, through the 70s and the 80s, there was a lot of natural history and a lot of youngsters really interested in, like me, I was so interested in turtles and snakes and all that. And Raghavendra, Professor Joshi told me this story. I don't know if he was supposed to. He said you nearly didn't get through the interview because you were too passionate about turtles. And I could see exactly where that concern came from for you as faculty, and the training that you provided us to think more like ecologists and evolutionary biologists, right? It was stop thinking about the animals as animals and think of them as models and think about the themes of your research and all of that. Now, that was a shift that was absolutely required for us at that time. 30 years ago, we needed to hear that and needed to think of our animals as models and points on a graph. We needed to change our thinking. 30 years later, in fact, I think the pendulum has swung a little bit in the other direction, where we need to tell people, like, go back to the field, spend more time observing animals, be more, be more passionate about the natural history. And I'm not saying that that people present here and young students are not. I'm sort of like not making any accusations, but in general, in the larger community, I feel like I'm almost now beginning to reverse the narrative that I heard from my mentors, and that over the first 10 years of my mentoring students, that was undoubtedly my own narrative. It's like, yeah, great, I get it that you love frogs or trees, but what's your question, right? And now I'm saying I get it that you have a great question. What do you love?
No, but Robert MacArthur has very nice advice. He says, love your natural history, but do not think that understanding the reason for patterns in nature takes away the beauty from nature.
Right. Absolutely. Absolutely. I'm so glad that we can invoke MacArthur. Great influence in all of my work. I hope that, I guess like I hope the people who've been here do have questions and are thinking about it, and I'm hoping that there's that whether you ask those questions here or not, that this serves as some kind of an opening for different sorts of discussions. I mean, whatever sort of little thought that it might have sparked in your head, I'm really happy to use this really as a starting point to chat about how our lives in science progress. Yeah, and wouldn't do anything else for if I were offered over and over.
I have a question, Kartik.
So good.
Yeah, so like when you were shifting fields, like shifting your group of study from small mammals to turtles, was it difficult to be taken seriously by the turtle guys since you had done your PhD with small mammals? And
see, in most fields, I would say the answer is yes. But the international sea turtle community is actually a very remarkable one. I was sort of like taken, under the wing by like a half a dozen different people at the first international conference that I went for. Like I said, Brian Bowen, who is this really well-known global researcher. Jack Frazier, who of course, extremely well-known in the turtle world, but was also at one point Nico Tinbergen's student and was one of the first people to study Aldabra tortoises. These people were like, oh, great, you don't want to study small mammals anymore? Come join us, right? And people were willing to offer me, help writing my proposal, send us like material whenever we asked for, and people in my own generation, like there was a bunch of people that those days there was not so much on the internet, so I wrote out to people looking for postdocs who turned out to be, my age and looking for postdocs themselves, and we were like, oh damn it, we're all competition, but people like Brendan Godley at the University of Exeter, and others in the US and Australia. In the first few years would become like first conference or two would become really close friends. So I would say within like my months of making that decision and going on to my first conference, which actually was really a seminal moment, I just felt like I was part of the community. So it was a very interesting, it was an interesting, valuable, and enriching experience for me. But I think it doesn't really answer your question fully, because I think that this doesn't easily happen in all transitions, which is, I'm not discouraging people from making them at all. I did, and it enriched my life in being able to bring everything I learned from sea turtles into biogeography, and everything I learned from biogeography into sea turtles, and then all of that got mixed up in some kind of marine biology interest. And so those interweaved threads have actually been really great for me. So I do encourage it, but my own personal experience with getting into the sea turtle community was yeah, it was quite remarkable. Forget about the first conference, that 97 workshop that I went for when I was a PhD student, and I wasn't even thinking of sort of formally working on turtles. They were like, oh, all right, great, come join us, like sit, tell us what you think. You used to work on turtles in Chennai many years later. So it's an incredibly warm community. And I think we're quite proud of it. I'm rambling again, but the other thing I should add that really attracted me about the Sea Turtle conference, the annual symposium, which for a group of 7 species, it attracts 1,000 people especially when it's held in North America, and 600-700 people when it's held in other parts of the world. But it's not just academics. It's like maybe a third or 40% are academics, but the rest are all conservation practitioners, people from government, even folks working in community groups and so on. I mean, those who can afford it. And for the longest time, we had one of the largest budgets for international travel, so that people that couldn't afford it would be brought over on student grants and support grants. So a fairly diverse group of people from different parts of the world. And so there's being at a conference that's not just a pure academic conference and had all of these people from these different sectors was also something that I found really appealing about the community. That's why I hung around for 20 years. There are other stories about the Sea Turtle Symposium that I can't tell in this forum. Those will have to be done over those cups of warm tea that I was talking about earlier. Harish?
Hi, Kartik. Thanks for the talk. I just wanted to ask about the age of the grasslands, if there's any consensus that you've come to. And it's interesting how from doing phylogenetics, you're able to tell something about the age of ecosystem or sort of thing like that.
I don't precisely know off the top of my head. And the thing was that the thing is that the analysis that we did earlier, we were not really looking to date the grasslands, right? But I think if we went back and reconstructed, conducted some analysis based on those trees that we do have now, we might be able to infer where some of those, what levels of divergence those early open canopy frogs appeared at. I think it's at about the clades diverge, if I'm not mistaken, in the range of about 15 to 20 million years, and I think that those open canopy groups also appear in that 10 to 15 million year time frame. If I'm not mistaken, that's about the same date that Ishan Agarwal puts on the lower elevation, grass open, habitat adapted species that he was working on. So we're all converging on that time frame, broad, big, giant, confidence intervals, which is not misaligned with what people know about the expansion of grasslands as a biome at a global, right? So let me say that at this point that the phrase that I would use is that sounds about right, but I think it'll need some more targeted sort of analysis to put a final date on it.
Hey, I have a question regarding what are the morphological differences between frogs in these open habitats and the ones in say the forest?
It's mostly the hind limb length.
Okay.
And yeah, I can of course I will have this is all Vijay's work and analysis, but I will have some figures that sort of show the differences between forest adapted frogs and grassland adapted frogs in terms of their hind limb length and other sort of I mean, I also look at it across like about the they separate on on a couple of on the principal component axis as well. So there's like a combination of traits that they vary across. But I think that the key one is the hind limb length. So.
Yeah, I think if people don't have any comments or questions, we can end this session. Thank you for this very nice talk, Kartik.
Thank you, Tanveen, for asking me. It was an absolute pleasure. And thank you, guys, ESS. Tanveen, of course, coordinating this talk and series, but all of ESS for the amazing work that you guys have been doing. Yeah, really. As I think Maria said earlier today, we sort of pride ourselves on being, the kind of department that we are, and thanks ESS for facilitating and enabling that. I'm just delighted to be able to sort of like, even give a talk of this nature, which I'm not sure that I would have been able to or even wanted to do in other in other forums. So thanks everyone.
Thanks, Kartik.
Thank you.
Yeah, thank you, Kartik.